Hello everyone, my name's Jim Lovell. The presentation I'm about to give is going to be a little bit different probably to anything else you'll see at the meeting. It certainly has a very long title, but I hope you'll find it interesting and maybe it'll get you thinking a bit about your research and interests and how they may be adapted to other applications. I'd like to thank the organisers for the opportunity to speak, and I really appreciate the opportunity to pre-record this, as it will be 4 o'clock in the morning, I guess, in Tasmania when my talk is on. I am planning on being there and will be happy to answer questions at the end. So I thought I'd better give you a bit of a background on myself. I've worked with a few people who are attending this conference in the past, notably Barney, Carl and Mark, but most of you have probably never heard of me, either that or you've turned up expecting a talk from a 94-year-old astronaut. My PhD was done at the University of Tasmania on strong gravitational lensing and monitoring it, and that was using the Australia Telescope Compact Array. There's a nice picture there of the Mount Pleasant Observatory in Hobart, 26-metre antenna. That's the gravitational lens I worked on, 1830-211, it's a 1 arc-second double, and quite variable AGN. And the other big thing I did during my PhD was a gravitational lens survey in the Southern Hemisphere with the compact array. So following that, I did a postdoc in Japan at the Institute for Space and Astronautical Science, and that was on VSOP, the Space VLBI mission, which had just launched shortly before I got there. And that was a lot of fun. And after that, I went back to Australia and had a job with CSIRO until 2006, and that was supporting radio astronomy activities at Tivimbilla, DSN tracking station, and also doing my own research, and that was mainly in AGN and focused mainly, especially towards the end, on interstellar scintillation. And that's probably the most relevant to the discussions here, so I'll briefly talk about that work. So AGN scintillation, so we're talking about refractive scintillation, and we did a big survey called the micro-arc-second scintillation induced variability survey, or MASSIVE, with the VLA, and we did use it in a non-standard way. As you can see there, we had the array pointing in 5 different directions at once, and that was to get good sampling of a bunch of as many quasars as we could to see what their variability was like on timescales of hours to days. There's a picture of our collaboration, all except Tapio, who couldn't be there for this meeting. There's JP Marquardt, of course, who I'm sure would have loved to have been at this meeting. And then Lucina, Dave, Haley, Barney, and Tapio, who wasn't there. And the survey revealed that there's a strong galactic latitude dependence on fast variable AGN, and that really established interstellar scintillation as the mechanism responsible for the short period variability and not intrinsic, as was still quite a strong debate at that time. And it resulted in a nice catalogue of variable AGN that scintillate that has now been followed up quite extensively. So following that, a job came up back in Tasmania, which I jumped at, and that was to do with VLBI, but geodesy, and the program called AUSSCOPE, a government-funded program to build 3 new radio telescopes across the country. So that was my job for almost 10 years, and initially building an array of 3 new telescopes across the country. You can see where they are there. There's one in Hobart, one in Western Australia, and one in the Northern Territory. 12-metre antennas, and they were dedicated to geodesy and astrometry. And they're still going and doing nice work. So moving on now to 2017, as I was working in AUSSCOPE, it became more of a job of administration and management than research. And so I decided it was probably not for me anymore. So I left that job and formed my own little company of one person called Astro-Acoustics. And that's focused on 2 things. One is the adaptation of radio astronomy techniques to bioacoustics, which is what this talk's about, and the development of a new recorder and related research associated with that. And the other thing I do is provide a consultancy on things to do with VLBI, telescope operations and automation, which I've had quite a lot of experience with. In 2018, I got involved with a citizen science program called Nature Trackers for Tasmania, and that came out as a result of participating in one of their citizen science projects called Where Wedgie, which is when people go out once a year for a few days and look for wedge-tailed eagles, which is a threatened species in Tasmania. And not very well monitored, so to see how the population is changing over time and if things are improving or not and what the threats are. So the data came back from that for the first year and proved to be quite problematical, and the organiser of the survey was looking for someone to help with the data, and I thought that looked like fun, and that's how I got involved. As a consequence of that, I met some biologists, met some biologists, and one in particular, Stuart Newson, from the UK, got me interested in acoustics and bats, and now that's led ultimately to a new citizen science project within Nature Trackers called Call Trackers, which I'm running, and that's looking at surveys to find threatened species or potentially endangered species in Tasmania and to monitor them. And got me thinking about radio astronomy and bioacoustics and the similarities there are when you think about microphones instead of radio telescopes and sound instead of light. You'd think all of the techniques you use in interferometry would be transferable to bioacoustics, and the advantages are, of course, that you can reduce the noise by doing cross correlations, and you can locate and map things from where the signal's coming from, and you can also turn it around in VLBI, for example, and work out the positions of your locations to high precision. So one of my first questions was, do biologists do audio interferometry? And do they use multiple microphones to do that? And the answer to that question is yes, sometimes. However, it's not easy. Localization of audio signals in bioacoustics has proven to be quite useful, but very challenging for biologists. There's a nice review article there about localization by Reinhardt and co. And there's a quote there. We find that the labour-intensive steps of processing recordings and estimating animal positions have not yet been automated. And I took that up as a bit of a challenge. So if you can measure locations and movements, then you can start to look at things like the animal's territorial boundaries, habitat use and interactions. You can identify individuals because you have directions, and you can start to do some reasonable abundance measurements, which you can't do with a single microphone where you just know if the animal is there or not. And you've got phase information from an interferometer, so you can pick out sounds by concentrating on a particular signal phase. The core localization methods used by biologists fall into two categories, really. One is to use audio and measure directions and distances with single microphone recorders that are synchronized in time. And you can do a time delay of arrival technique for that. But you need to synchronize them to about a millisecond, which means you need GPS on each device and obviously a view of the GPS satellites, and that's not always possible, say, in forests or caves. The other techniques are using thermal imaging to find out where the bats or whatever it is are, but that doesn't provide identification, and because you've got no sound to use for IDs. The other is capture and tag and then radio tracking. That's time consuming and expensive and invasive, whereas audio monitoring is not. So there are some advantages to trying to stick to acoustics field. So my next question was that what if you could do localization with a single recorder, and by having multiple microphones all connected together and synchronized because they're connected together on the same device, and can you use radio interferometry techniques in this field to do that? So one of the big motivations for this is shown on this slide, and when I started getting interested in all of this, I discovered pretty early on that of all the threatened species in Tasmania, and there's the order of, I think, about 50 or so, less than 15% of them are actually monitored. So since they were listed on the threatened species list, nobody knows has much idea of how they're going. And how do you prioritise your conservation efforts if you don't know the work you're doing or would like to do to conserve and protect these threatened species is worthwhile? How do you know the population hasn't improved? Or how do you know if the population has declined and it really needs urgent attention? And part of the problem is that long-term monitoring is very lacking, not just in Tasmania, but many places, because it's long-term and it doesn't fit in well with government funding cycles. So I was looking for ways of getting around this or improving this, and how acoustic monitoring might be useful. And making acoustic monitoring and directional acoustic monitoring more accessible and more effective through things like automation and multiple microphones and new technology, and taking advantage of people who are interested in this and citizen scientists so that anybody can use it was a high motivation. So we're starting with bats. There are two types of bat. There are megabats, which are things like fruit bats, and they eat things like fruit, and they're large. They don't use ultrasound. They don't call. And then you've got microbats, which do call. There are about 760 species worldwide, and they're carnivores and insectivores, things like that. Of the Tasmanian bats, there are 8 species in Tasmania, one of which is endemic. They're all insectivorous. They all dwell in trees and not caves. And they're microbats. They're all quite small. So that's one of them there, and that's somebody's hand in blue there with a glove on. So you can get an idea of their size. Of those 8, there are 3 forest bats, and they're a bit problematical because they have very similar calls, very hard to tell apart. And we're looking for ways of doing that, maybe finding some different characteristics in the call signature that will help us distinguish them. And just quickly, here's a plot that shows pulsars and bats in the same plot, possibly the first time we've seen something like this. The top plot shows Vela over 2 seconds and 11 pulses per second, of course, and the bottom one shows a bat in my back garden, and it also shows similar looking profiles at about a similar rate. So that's sort of quite interesting. So why bats? Tasmania's bats aren't threatened or listed as threatened, but they're also not monitored, so we don't really know. But we do know that they face quite a few threats, some of which are quite new. There are a lot of wind farms popping up around the state. On the mainland of Australia, there are diseases emerging which are affecting bats. There's loss of habitat in Tasmania due to forestry. The forest tree hollows are being lost, and there's loss of habitat due to climate change, and also potentially there are pesticides that are causing problems. Also, in other countries like the UK, quite a lot of money is spent on bats related to infrastructure development and site usage and protection of bats on various sites of construction. There are some significant data challenges with all of this, and they're familiar to anybody who's done any interferometry. For example, you collect phase, but there are phase rotation ambiguities if the microphones are more than a wavelength apart, and you need to recover delay. So there are some issues there that you have to deal with. Also, in this case, it's a near field effect for quite a lot of things that you might be trying to listen to, and you need to take that into account. The Earth doesn't rotate underneath the object you're looking at, so you need good UV coverage instantaneously. There are no natural calibration sources. You can't integrate in time because the signal's always changing and coming on and off and moving, and distance measurements are quite difficult, as it turns out. So I went through a couple of homemade recorders. This is the second one, the Mark II, and had 4 microphones. The first one had 2 microphones and was a nice sort of proof of concept that you could measure direction with a stereo pair. This one had 4 microphones and meant that I could do some more serious testing in direction measurements. So here's the data I'm going to show you from the Mark II recorder. You can imagine it as a badly behaved spectral ion pulsar, but it's actually a large forest bat. The time series is shown there. You can see amplitude against time, and you can see some variation in amplitude and call rate. And there's a picture of the bat itself. It's tiny. It weighs about 4 to 10 grams, and the overall size is about 10 centimeters for a big one. The top plot here shows the spectrogram, so we're looking at frequency and time, and amplitude is the colours, and each of these is a chirp from the bat, and you can see it going through a number of different modes. It starts off in its hunting mode, and you can use the time difference between the chirps to work out how long the bat is waiting before it gets an echo, and that corresponds to a distance. So when it's hunting, it's sort of looking up to about 13 meters into the distance, and then you can see the call rate increase as presumably it's found something, and now we're looking at something that's closer, about 8 meters, and then it goes into what's called a feeding buzz, which you can see in more detail down the bottom there. And now the time between calls is about 5 milliseconds, which corresponds to a distance of about a meter. And you can see the frequency drop and the call rate go up to its highest level, and this is presumably what the bat homing in on the insect, and it catches it sometime around about there at 3.6 seconds and eats it, and then it goes back into its hunting mode and continues on. So even just by looking at the call pattern, you can see something about the bat's behaviour. Here's an individual chirp from this bat, and there are two sections to this spectrogram. You can see a sweep in frequency from the bat from a bit over 60 kilohertz down to about 38-ish. The frequency sweep gives it a sense that gives it responses over a range of scale sizes, and the louder call at the bottom is mostly, I think, for range. And that would be at the lowest frequency because that's the largest scale size and sort of covers, I guess, bigger things. And then you can see on the right hand side what gets sent back, and this is what gets sent back to the microphone, not to the bat, although they may or may not be the same depending on which way the bat's flying. You can see there's probably a frequency dependence in that, and that there's a bit of a delay between the call and the response, which you can see more clearly in other plots. And the plot on the top right here is something similar. It's not the same call, but it shows a phase as a function of frequency and time for a chirp. And you can see the phase is nice and constant at the leading edge, which is the bat call, but is mixed for the reflections, as you'd expect. So here are the scale sizes for the call. So the bat is sensitive to scales from about 1 centimetre to half a centimetre in this particular case. And it's getting reflections mostly of sizes above 8 or so millimetres. And you can look at the delay also between the call and the response. And that gives you some idea of the path length, or the additional path length that the signal's taken to get to you from the bat, and tells you something about the environment that the bat is calling in. So here's that data again, and there's a spectrogram which you've already seen. This bit down here is a sky map, and it's going to show you the direction the sound is coming from. Imagine you're lying on the ground with your head pointing north, so your head is and that's the northern horizon, that's west, east and south, and zenith is straight up. So I'm just going to step through this, and you'll see in a moment I'll just pause that and drag it back. You can see now every time we go over a chirp we get a new point on the sky map. You can see the bat starts off toward the north at about 20 degrees elevation and is heading, in this case, pretty much overhead, which is good for us. And now you can see those the call rate increasing, and just as the feeding buzz starts, you can see the bat change direction. So it's picked up on the insect and it's chasing it, and it catches it around about there as it's finished turning the corner, and then it goes back into its search mode, and off it goes off roughly to the west, a bit over. So that's what you can do with a 4 microphone recorder. However, the technique is still quite labour intensive and needs quite a bit of automation. So we're finally up to secondary spectra. Here are 2 taken from that data set, one in hunting mode and one when it found something. So those are the spectrograms of the chirps at the bottom. You can see some differences in when the reflection occurs. It's quite a big gap in that one, for example, and you can see the corresponding secondary spectra. They are certainly not symmetrical, and there's looks like some interesting stuff there, but this is as far as I've got so far. This is quite new, at least to me. So looking promising, I guess you could say, at this stage. So what next? The plan now is to develop a usable production-ready device that we can do some serious prototyping and testing of, and to build a more robust processing technique, and make an automated pipeline work better so that anybody can use this device or these devices. And we also I'm still looking into other astro-type processing analysis techniques, particularly secondary spectra, using amplitude and phase. Those plots were just amplitude that I showed you today. Maybe secondary spectra can also help with identification of species, especially the problematical ones, and maybe even bat behaviour, given you can start to look at the differences and between the left and right hand side of those secondary spectra. And maybe even use the equivalent of dispersion measure in audio to estimate distances to the calls. So now we've moved on from the Mark II recorder to a real prototype, and we're using a company called Definium Technologies who are partnering us with this. They're based in Tasmania and do complete end-to-end instruments for industry and research. They have their own manufacturing facility in Tasmania. The designs are now being finalised, and we're hoping to get prototypes out by the end of the month and start debugging shortly after that. So what will the prototype look like? The idea is to build into it as much as possible, more than we'll eventually end up with, to try out all of the possible technologies. The general idea is that we have a hub unit which has a few more features, including the ability to communicate with all of its nodes around it, which have overlapping audio coverage, but less stuff on them. 4 microphones each and some sort of communication back to the hub, basically, and storage and batteries. Each one will have 8 microphones. The arrays will be configurable, and as will the microphone types. And we'll be recording up to frequencies that cover bats, as well as regular audio. You can read the other things there, but the idea is really to try out all of the possible techniques and develop something that's going to fulfill all of the possible uses that we can think of. Following development of the prototype, there will be extensive field trials and consultation with real biologists, and we'll publish what we find and hopefully develop something that can be usable for anybody who's involved in this sort of thing. So if any of this has got you interested, there's way too much work for one person, and it would be great to have people with skills that I don't have involved, especially if you know a lot about scintillometry. I don't know who that could possibly be. So if you're interested and have some ideas, or think you are interested in helping in some way, please get in touch with me and I'll be really happy to talk to you. So thanks, and I'm more than happy to answer questions.